Have you ever wondered what animals can see? Like your cat or your dog, or a fish that lives underwater, or a bat that flies in complete darkness, or even an eagle that has to catch a teeny tiny mouse on the floor? Do they see the same thing as you do, or do they have a vision adapted to where they live, what they eat, in other words, their environment? I'm Alexa Sadier, a research scientist at UCLA, and this is the kind of question I'm interested in. What animals can see and how their vision evolve in adaptation to where they live, and how can we find big evolution principle by using these results. And you might be surprised if I tell you what I study, because I don't study all the animals in the world to answer this question. I'm studying a group that is diverse enough so we have a lot of different species that live in a lot of different kind of environment and eat a lot of different things. And this group is bats. And you might be surprised because people have so many misconceptions about bats, but they are also indeed really diverse and have everything we are interested in. So first, diversity. Just look at them. Some of them are red, some of them have big ears, some of them have a lot of different colors. So just by looking at these species I show you here, you see how incredibly diverse they are. But even that is just a subset. It's only 30 species. That is a small subset of the 1,426 bat species that we have in the world, which is a fifth of mammals. And as I said, we're interested in vision, we are interested in what animals can see. And if you look at these bats again, you can see that we have the diversity in terms of vision as well. I mean, in terms of their eye size. Some of them, like this fruit guy, have very, very big eyes. So that are very, very big compared to his head. But others have eyes so tiny that we can barely see them. And if you look at the other bats, you can see that we have a lot of different eye types, different colors, etc., etc. As everywhere in science, every time we start with an observation. And by looking at these bats, we can see that we have this diversity of eye types. So maybe we have a diversity of vision. And maybe this is because these bats are adapted to their environment. So where do they live? Because if they are adapted to their environment, they might live in different places. And so to do that, let me show you this map. So you can see that we have bats almost everywhere in the world, except in the polar region when it's white. And this map also tells us something else. The color of the map shows us the number of species of bat you can find. So when it's red and yellowish, it means that you have a lot of bat species here. So if we're interested in looking at vision evolution in a lot of different bat species, of course, we want to go when we have a lot of bat species, which is in the reddish and yellowish region. Meaning that when I want to study bat, I go to the tropics. And here in yellow, you can see the different field sites that we have. So we go to the Caribbean here in the island and the mainland. We also go to Africa here in Cameroon, a tropical area as well. And we also go to the Philippines. And if you are not convinced that just by going there, we are hitting a lot of different environment, let me bring you into the field so you can see how it looks like. For example, when we go to Belize, we go to these wonderful, magnificent forests that are really dense, and bats roost in these forests. But as you can see here, we also have some Mayan temples. So it's really, really exciting. It's kind of Indiana Jones style fieldwork, because bats also root sometimes in some of these temples that are not excavated. So we have one area here. When we go to Puerto Rico, we also see some big, deep jungle forests, and we have different species of bats here compared to Belize, which is the mainland. So we have another type of habitat, which is the island forest here in Puerto Rico. When we go to Puerto Rico, again, in other places, we have some cave habitats. So it's another type of habitat here, a cave, a deep cave when we find bats as well. And finally, when we go to other places such as Trinidad, we have cave underwater, we have forest, but we also have houses in which we have some bats that fly. So just by going into these different places, we have all these different habitats where bats live, and so then we can study how their vision adapts to that. But as I said, something we can consider as well is food, to see if bats are also adapted to where they eat, because they are foraging, they need to see what they eat very well. And just by going there, catching bat there, we also have the full different range of diet that bats can have. For example, if you look on this picture here, when I catch some bats, I can understand what they eat. If I catch one bat that has this very, very elongated snout that you see here, and if this bat is also covered with pollen, it's likely to be a plant bat. The other bat you can see here is the vampire bat that eat blood. I caught this one on a chicken on a tree. And as you can see, some of the finger of that bat is covered with blood and also its face is because it's a vampire bat that eat a lot of blood. Now, you can see as well that we can catch some insect bats that fly at night and are catching a lot of insects. We can also find some fish bats because some bats are fishing. And so you can see on this picture a bat just arriving and catching a fish in the water. We also have bats that eat animal, omnivorous bats. And we also have bats that eat fruit. This is a teeny tiny fruit bat that I caught in the forest. And sometimes before releasing them, we just give them a little snack. This one is from Belize. Okay, so by looking at these different places, these different bats and the diversity, can we now ask the question we want to ask in this talk? So the first question that we have is, what do bats see? Do they see a lot of different things? Do they all see the same? We don't know, so what do they see? And we have seen that they live in a lot of different places and eat different things. Do their vision relate to where they live and what they eat? Do we have an adaptation to these different lifestyle and environments? And finally, the other question we can ask is, what can it tell us about the evolution of species in general? Can we find big principles just by studying that vision that will be applicable to a lot of different kind of things? So before digging into the results, I just want to give you a little primer about how do bats in general and mammals see. So bats see through their eyes, so it's exactly like us. The light just hit the eye and hits the back of the eye. And it's very important to understand that because in the back of the eye, we have the retina, which is the tissue which is sensitive to light. And in this tissue, how do this tissue sense light? It senses light through photoreceptors, the rods and the cones in green and blue here. And these photoreceptors are the cells that are able to sense the light and to relay this light message through a couple of cells and neurons into the brain that will reconstitute the images. As I said, we have two types of photoreceptors. Why do we have two types? We have two types because they are sensitive to different types of light. First, we have the rods that are sensitive to very, very dim light. So they allow us to see during the at night or in kind of a darkness with very, very low light. And we have only one type of photoreceptor, one type of rod. So that's why we see in black and white. During the day and when the light is at dusk and dawn, so low light, we have the cones that are active. And we have three types of cone, and it allows us to see in all the different colors of the visible spectrum. How? So we have some red cones, green cones, and blue cones. And if you don't understand why having three types of cone allow us to see the different colors of the visible spectrum, just think about your computer screen, or if you try to modify a picture on your screen. You always play with the red, the green, and the blue channel. And by changing the amount of color that you have, you are able to reconstitute the different color that we see. It's the same as our brain. Our brain do that in humans. We have a red cone and blue cone and a green cone. And by playing with the different intensity of this cone, we're able to reconstitute the color of the rainbow. And for the rest of the talk and for the bats here, we will focus on the cone. So we won't focus on the bat vision in general, but we will focus on color vision in bats. So what do bats see? Do they also have red, green, blue like us? Actually not. The study that have been done on bat color vision and on cone, etc., have revealed that bats seems to have two types of photoreceptors. S cones and L cones. S cones is responsible for UV light vision, so a lot of bats can see UV, and L cone is responsible for greenish light vision. So what we think, these studies have been done on a very small subset of bat species, only 10-ish bats, species of bats, have shown that maybe the ancestor of all bats have UV and green vision, and that maybe some bats have lost some vision. But of course, when you think that we have 1,426 species of bats, we don't really know at all how it evolved in a lot of different things. So this is what we try to look here. But in fact, what does it mean for the bats to have only two types of cone for color vision? This is a picture how we will see it with three different cones. And this is what bat would see with two different colors. So they would see something that would be kind of close to what colorblind people would see, people that lack the red cone in human. So they would see that. So of course, if you think about being a bat and seeing this, you can see that it can have some implication in how you will discriminate food on the foliage, etc. And if you lose one of the two cone, you will see basically black and white. So a better understanding color vision at this point for a bat, for the point of view of the bat, and also for the point of view of evolution, can, help us to understand what's going on. So how will we study the presence, absence of these cones and the color vision? How we will do that in the lab concretely? So we want to focus on the cone themselves, but we will focus on the pigments that sense light into the cone. If you remember what I said before, the cones are able to sense light and to relay the signal into the brain. And this is done through two types of protein, the opsins, that are the pigments, the protein that sense light inside the cone. We have two of them. The S opsin that is responsible to sense UV light. In S cones, you have S opsins. And the L opsin that is responsible to sense green light. In L cone, you have green opsin. And we won't only study the presence in this protein in these bats. We will study the presence of this opsin at the three levels of gene regulation that we can have. We'll study the presence of the gene, the S opsin gene, then the L opsin gene in the genome of a lot of bats. We will see if this gene is expressed by checking of the presence, absence of the RNA of these genes. And we will also check if a protein is produced at the end. So do we have an S opsin and an L opsins to make an S cone and an L cone? So keep that in mind, it's really important. We will study all these three levels of regulation here. Okay, so how we will do that in the lab? How we will study these three levels of regulation? So to do that, we will do that on 55 species of bats that span the variation in terms of environment and the variation in terms of the diet that we can find in these bats. And we did that in field species, so in species that we caught in the wild, but also using museum specimens for the protein at least. And I will talk a little bit more about that in a minute. And so let's start with the protein first. How do we study the protein? So to do that, we take the whole eye of the field species or the museum species, and we carefully dissect this eye to have access to the retina. So remember, the retina is at the very back of the eye. So we open the eyes and we have access to this tissue, the retina in the back, where we can find the photoreceptors, where we can find the cones, and so the opsins. And on this retina, we then use an antibody that will recognize specifically either the L opsins or the S opsins. And just by putting this antibody on the retina, we're able to see if we have or not the S cone and the L cone. How? Because the antibody will recognize specifically the opsins, and when you have a lot of opsin in a cone, you will have a dot here that you see in green for here the L cones. And so just by doing that, we're able to see the presence, absence of the cones in these bats. And we have done that on field species, so in species we caught in the wild of this dissection, but also, which is kind of pretty cool, on museum specimens. And some of them are really, really old, like 100 years old, all of the nests, which is super interesting because it just shows us that we don't necessarily need to go in the field and catch species where they are and, use wild animals to do this kind of study, but we can also use museum specimens, which just show us how important museums are and what resources we can get from this museum. So this is what we do for the protein detection. How do we detect the RNA, the mRNA, so the expression of the opsin gene? So to do that, we take the whole eye of a bat and we sequence all the mRNA coming out of these eyes. And we were looking for the presence of the L opsins and the S opsins in this subset of mRNA that we have in the eyes. And finally, we look at the presence, absence of the S opsin, of the L opsin gene by PCR and sequencing in the genome, because for a lot of these bats, of course, this is a non-model species. So we don't have the genome. We have the genome of only few of them, and we probably have more genome in the future. But right now, we just can't just go on the database and look for a gene on the genome because it's just not there. All right, so we have the setup to study protein, mRNA, and DNA. So what are the results? What do bats see? So let's start with the protein first. What can these flower-shaped retina, what is the study of the protein can tell us about bad vision? It can tell us a lot more than just the presence absence actually. So we have this flower-shaped retina here. You have an example of an insect species. And we were looking for the presence of the L cones and the S cones. As you can see for this species, we have the presence of the L cones and the S cones. We can see the dots for both things. But it's not the only thing we can see. By looking at that, you can also see that we can assess the density of the cone that we have. Just by counting the number of dots that we have by square millimeter, we can see if a bat would have more cones than another one, or if we have more L cones than S cones. And it is very important for bad vision. Just again, think about your camera. You have a number of pixels. The more pixels you have, and the best the resolution of the camera would be. It doesn't translate exactly like this for vision, of course, it's more complicated than that, but just keep in mind that changing the density of cones would have kind of the same idea, the pixel of your camera. The vision would be different, the things you would be able to see will be different in your brain. And so just by assessing the difference in terms of cone density between bats will help us to understand better, to see if bats have a different vision. So let's do that of a lot of different bats. So here you have a subset of what we have been doing in many bats that have different diets and that live in different environments as well. And here the diet is indicated with the little insect or the flower or the fruit, etc. So the first thing we realized is that we were able to detect S cones and L cones in many species of bats with different diets. So that's interesting. But then we realized that we don't detect S cones everywhere. Some species have lost S cones and so have lost UV vision. So here we have some species that have the two color visions, so that are able to discriminate colors, and some species that have only L cones, so they are only able to see one color, and so that probably see kind of in black and white. So we have these two first different types of bats, which is interesting. They don't all see the same thing. The other thing we can see is that the density of cone varies a lot between species. We have some species here that have very dark retina in the little cartoon that I have here that have a high density of cone, whereas others, like for example, Carolia soweli here, have less L cones and even less S cones, showing that the vision capabilities between bats are different. And this is probably because these bats don't need to have the same vision capabilities to be able to forage or sense food, etc., etc. So we have this huge variation between bats. It's not fixed. All the bats seems to be different things. And we also have less S cone than L cones when we have both, which is again interesting because the only pigment that we lose is the S opsin. So we lose the S cones in some bats. And when we have both, we also have less S cones, meaning that maybe it's not that important, but it's just something that is gained or retained in the bats that need to have this combination of color and to see UV vision. Okay. So we lose the S cone in some bats, and what is the mechanisms behind the loss of the S opsin genes? So to do that, we will look at the conservation of the DNA, the mRNA, and the protein, the three levels I talked about before, just to see if it can give us some insights about how do we lose a trait and these genes in bats. So first, as I said, the L opsin genes and the L cones are present in all bats, seems to be there in all bats. And this is exactly what we see here. We can find in all the bats that we study here, it's a subset of the 55 species of bats we study for clarity. And that you can see is we indeed have a conservation of the DNA. We have a conservation of the mRNA expression and the protein. They are present everywhere in the green cones, in the L opsin gene, which is what we expect. Now, when we look at the S opsins, the picture seems to be completely different. We have a loss of the S opsin genes, but a different level of regulation. So if we look, for example, at these species, we have what we would expect when we first think about it. We have lost the S opsin genes. We don't have an expression, of course, in the mRNA, and we don't have an expression of the protein either. We have a complete loss of the S opsin genes in these species. Now, if we look at another species, the picture seems to be different. We can still find the S opsin gene in the genome, but we don't have the expression of the mRNA, and of course, we don't have the expression of the protein. So for this species, we still have the gene in the genome, but we have lost the other level of regulation here. And we have another case when we can find the gene in the genome and the mRNA is expressed, but we can never find a protein. So here we have lost this S-opsin at another level of regulation. We have the DNA, we can find the mRNA, but we will never have a protein expression of the gene. And this is really important because just by looking at these three levels of regulation, we're kind of catching evolution in action. We can see that when we lose a trait here, when we lose the S-opsin, we don't necessarily lose everything at the same time and everything from scratch. We don't necessarily lose the gene, the mRNA, and the protein. We have different steps where we can lose different level of regulation. Now, as I said, we are trying to relay these losses to the adaptation to environment and food. What's going on? By just looking at these different species here, can we find a link between the loss of the S-opsin and the adaptation to different environment and food? So to do that, we did like a test to see if we can link the diet or the roosting capabilities, the environment, to retaining S cones or loss. So let me walk through this figure here. So we have this different condition. If we're on the 0 line, it means that we have no association with retaining S cones in these conditions. So for example, for cave roosting here in blue, we are on the 0 line here, meaning that we have no association between retaining S cones and roosting in a cave. On the other hand, if you look at the green and the pink violin plots here, we're above the line, we're near 5 here, meaning that we have an association between eating fruit or visiting plant and retaining S cones. So it means that here we have identified clearly that if you eat fruit or if you visit a plant, you are more likely to retain S cones. So it seems that vision and the type of things that bats see is related to diet. It's not absolute. You remember I showed you also some a species that eat insect and have S cones. So it means that we might have other factor, but we don't have enough species here to identify that. And it also means that we need to do more studies to do that. Okay, so we have investigated a lot of things. What to conclude about bat vision and evolution in general? What we can get from these results? Well, first, these results, as I say, tell us a lot about bat vision. Because with this, we have identified two main types of color vision in bats. One type of color vision that only have L cones, and so these bats see probably in black and white during the day. And we have one type of vision with two types of cones, S cones and L cones. These bats seems to be able to discriminate colors, but it's not absolute. We might have different factors, but they might be able to discriminate color, maybe to find food, maybe because it helps us to see the food better, the fruit, for example, on the foliage, etc. So we have these two different types that seems to be very important and linked to diet. But as I said, it's not absolute. We also have insect bats that are able to see UV. So how can we explain that? Let me bring you back into the field because just by looking at bats in their environment can help us to understand that. Here, this is a video of bats that are flying at night. So if you just look at this video, you can see that bats are coming out of the cave. And this is very early at night. We try to set up the traps early to catch them because we really don't want to miss anything. And you can see that the light is still very high in the sky. The light level is still high, meaning that these bats fly out of the cave when it's not dark out yet. So maybe they do need to have a better color vision because they fly during the day. Now we also have some bats that come out of the cave when it's really dark, and maybe these bats don't need to have a good daylight color vision because the light is already very, very dim when they go out. So this is something we can investigate, and the only way to investigate that would be to study more different species. Now, a lot of bats are also endangered. Is it useful to really understand what bat can see to protect them? Actually, yes, because if you look around where you live, for example, you can see that we have a lot of light in the street at night, in the parks, etc. And of course, as you can imagine, you have bats everywhere in the world, so you have probably bats around. And so by understanding what they see, it can also help to protect them to find a light that won't affect them. For example, we do think that red light won't affect bats as much as white and green light, for example. So we can think about how to change the light that we have in the street or in the parks or in different areas to be better for bats and to help them, to protect them, etc. So we have seen a lot of things for bats. What about evolution in general? This study also helps us to better understand trait loss. Because when we think about evolution, we always think about new things that animals gain. We gain, for example, new abilities to be underwater. Some species gain new abilities to fly, but we never think about how we can evolve by losing things, which is something that has been very important in evolution. When you think about it, dolphins, for example, have lost their limb, or cavefish have lost their eyes in the cave. Here we can see that bats can evolve by losing UV vision. And it's another textbook example of how species evolve by losing something. And our studies also show us how we can better understand trait loss and how to study trait loss. Because a lot of studies focus on the morphology or only the DNA to assess stress loss. When we have a trait that is controlled by a gene, which is the case here for S. obscens, where we tend to look only at the DNA to assess the presence, absence of this trait. But what we have shown is that we can't only do that. Sometimes we have the DNA, but we don't have the protein. Sometimes we have the RNA and we don't have the protein. So our studies show that it's important to look at the whole process to really understand what's going on. And it also tells us a lot of things about possible lost and regain, etc. Because you can imagine that if you lose only the RNA, maybe we have a possibility to regain this trait later if we need so, if we have kind of a hotspot of evolution when we lose or regain trait easily. So just by looking at all the process, we're also in a better shape to really understand what's going on in evolution and the possible mechanisms. And our study also shows how important it is to study a lot of different kinds of species and not only one. Because if we study a subset of species, we can be like, okay, bats see in two colors, or these species have this trait loss, etc. And our study show that it's way more complex and diverse than we think it is. So to really understand how species evolve, we need to study a lot of them. Okay. So what are the main message of that talk? I think I said already everything, but just to sum up, first, we have shown here that bat color vision is adapted to diet and other conditions and is way more complex than we think it is, and that we really need to study all the bats to really have the big picture. We have also seen that by studying a lot of different species and this kind of trait, we can catch really evolution in action. We can see what's going on. Species have evolved but are still evolving, and especially in this world that is changing with climate change and everything, it's really important to understand how species evolve to protect them and to be able to understand what will be going on. And finally, it's really important to study biodiversity as a whole because it can help us to reveal general concept about evolution and to really understand how species live in this world and evolve, etc., etc. For this study, I want to acknowledge a couple of people. So of course, the people of the Sears lab I'm in, my boss Karen Sears, and all the people that participated in this work. I want to also thank the main collaborators because this work has been done in collaboration with many people in an NSF grant. And so I want to acknowledge the main collaborator on that project. And also I really want to acknowledge all the people in iBiology and my coaches to help us to shape this talk. And thank you all for your attention.